Assalamu alaikum. I am Dr. Refat Tabassum, Associate Professor, Department of Biochemistry, Bashundhara Ad-din Medical College, South Keraniganj, Dhaka. Today we are going to discuss about water-soluble vitamin, vitamin B12, which is known as pernicious anemia vitamin. Now vitamin B1, it is also known as anti-beriberi vitamin. Thiamine is known as vitamin B1 or aniuretic hydrochloride. Dietary source. Cereal grains are the most important dietary source of thiamine in the diet. Of the cereal grains, the whole grain contains more thiamine than refined grains. Thiamine is found in outer layer of the grain as well as in the germ. The main sources are brown rice, whole grain flour, pulses, oil seeds, nuts, cauliflower, potato, oranges, yeast, liver and egg. The RDA is 1 to 1.5 milligram per day in adult normal. Function. Thiamine pyrophosphate acts as coenzyme for enzyme pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, transketolase, which catalyze the reaction of the pentose phosphate pathway. Thiamine pyrophosphate acts as important role in transmission of nerve impulse. Deficiency syndrome. The deficiency of the vitamin B1 result in a condition known beriberi. It is most seen in the population consumption exclusively polished rice as staple food. Beriberi is categorized into three types: dry beriberi, which neuromuscular symptoms are predominant; wet beriberi, cardiovascular manifests appear; and infantile beriberi, like wet beriberi. Wernicke-Korsakoff syndrome also occur in the thiamine deficiency in alcoholics. Now dry beriberi. Dry beriberi manifests as peripheral neuritis. The muscles become progressively weak and walking become difficult. The patient become bedridden and may die if untreated. Wet beriberi. The wet beriberi is characterized by edema of the leg, face, trunk and serous cavities. Breathlessness and palpitation are also present. The calf muscle are slightly swollen. The systolic blood pressure elevated and diastolic decreased. Heart become weak and death may occur due to heart failure. Infantile beriberi. The infantile beriberi is seen in infant born to mother suffering from the thiamine deficiency. It is characterized by restlessness, sleepiness, vomiting, convulsion and abdominal colic. These symptoms are due to cardiac dilatation. Death may occur suddenly due to cardiac failure. Biochemical changes in vitamin B1 deficiency. Carbohydrate metabolism is impaired and pyruvate is accumulated in tissues. Pyruvate cannot cross blood-brain barrier and enter into the brain. The alteration of the blood-brain barrier permitting pyruvate to enter into the brain. Now riboflavin, vitamin B2. The flavin, comes from the Latin word flavus, mean yellow. It is the common name. The rich sources are milk and milk product, meat, eggs, liver and kidneys. RDA ranges from 1.2 to 1.7 milligram per day. Functions. Riboflavin is the precursor for coenzyme FMN and FAD. FMN is associated with electron transport chain. FAD is associated with certain enzyme involved in carbohydrate, protein, lipid and purine metabolism. Deficiency symptoms. Symptoms associated with riboflavin deficiency included glossitis, that is inflammation of the tongue, angular stomatitis, inflammation of the angle of the mouth, cheilosis, inflammation of the gum, seborrhea and photophobia. Now niacin or nicotinic acid, which is also known as pellagra preventive factor. Niacin is referred as vitamin B3 because it was the third of the vitamin B to be discovered. The word niacin is coined from the letters of three words: nicotine, acid and vitamin. Niacin is a nutritional term used to refer the vitamins nicotinic acid and nicotinamide. Coenzyme form of niacin is nicotinamide is incorporated to the structure of two coenzyme NAD and NADP. Dietary source: animal products, liver, heart, kidney, chicken, beef, fish, milk and egg. Plant sources included whole grain, cereals, pulses, peanut, sweet potatoes, leafy vegetables, carrots, etc. Synthesize: we synthesize niacin from the tryptophan. The essential amino acid 60 mg of tryptophan equal to the 1 mg of nicotinic acid, and RDA is 15 to 20 mg. Biochemical functions: the coenzyme NAD and NADP are involved in a variety of oxidation reduction reactions. The important enzymes where NAD acts as coenzymes are glyceraldehyde 3-phosphate dehydrogenase, pyruvate dehydrogenase complex, alpha ketoglutarate dehydrogenase complex. The important function where NADP acts as coenzymes are glucose 6-phosphate dehydrogenase and HMG-CoA reductase. Deficiency symptoms: niacin deficiency result in a condition known as pellagra. The disease involves skin, GIT and nervous system, and it is referred as 4 Ds: diarrhea, dementia, dermatitis and if untreated, death. Here is a picture of dermatitis. Now pyridoxine, vitamin B6. It represents 3 compounds: pyridoxine, pyridoxal and pyridoxamine. Pyridoxine is a primary alcohol, pyridoxal is an aldehyde and pyridoxamine is an amine. The active form is pyridoxal phosphate. Dietary sources: animal, egg yolk, meat, fish, and plant source are wheat, corn, cabbage, roots and tubers, and RDA in adult 2 to 2.4 mg and in pregnant and lactation period 2.5 mg. Function: pyridoxal phosphate participate in the reaction like transamination. Aminotransferase enzyme needed to broken down into amino acids are depend on the presence of pyridoxal phosphate. The proper activity of this enzyme are crucial for the process of moving amino group from one amino acid to another. Decarboxylation: pyridoxal phosphate dependent decarboxylases plays a role in biosynthesis of 4 important neurotransmitters: serotonin, epinephrine, norepinephrine and gamma aminobutyric acid. Trans-sulfuration: pyridoxal phosphate is a coenzyme needed for the proper action of the enzyme cystathionine synthase. This enzyme work to transfer methionine into cysteine. Deamination: deamination of hydroxyl group containing amino acids require pyridoxal phosphate. Condensation: pyridoxal phosphate is required for synthesis of the and condensation of the glycine and succinyl CoA of amino livoic acid by the enzyme alanine aminotransferase synthesis. Deficiency symptoms: neurological symptoms are depressions, irritability, neuromuscular weakness in short term deficiency. There may be significant long term side effects of peripheral neuropathy. Now biotin. Biotin or vitamin B7 is a sulfur containing vitamin B complex. It is shown that consumption of raw egg white causes deficiency symptoms of the biotin. Function: biotin serves as a carrier of carbon dioxide in the carboxylation reaction. The reaction is catalyzed by pyruvate carboxylase, where pyruvate is converted into oxaloacetate. Dietary sources: biotin is widely distributed in animal, plant and plant foods. The rich sources are liver, kidney, egg yolk, milk, tomato and grains. Biotin is also synthesized by intestinal flora. The RDA is 10 to 300 mg for adults. Deficiency symptoms: biotin deficiency is uncommon. The symptoms included anemia, loss of appetite, nausea, dermatitis, glossitis. The deficiency also causes depression, hallucination and muscle pain. Now pantothenic acid. Pantothenic acid, the Greek pentos mean everywhere. Its metabolic role is coenzyme A is also widespread. Dietary sources are mostly distributed vitamins found in plants and animals. The rich sources are egg, liver, meat and milk. The RDA is 5 to 10 milligram for adults. Function. The function of the pantothenic acids are exerted through coenzyme A. Deficiency symptoms: burning feet as symptoms may occur, and numbness of the toes and sleeplessness. Now, folic acid. The term folic acid is derived from the Latin word folium, means leaf, because it is mostly found in the green leafy vegetables. It serves as a carrier of 1-carbon metabolism during the several biosynthetic process. The active form is known as tetrahydrofolate. It is synthesized from folic acid by the enzyme dihydrofolate reductase. These are the chemical structure of the different tetrahydrofolate groups. Active center of the tetrahydrofolate. The N5 position is the site of attachment of the methyl group. The N10 is the site for attachment of the formyl and formamino groups, and that both N5 and 10 are bridge the methylene and methyl groups. These 1-carbon transfer reactions are required for the biosynthesis of the serine, methionine, glycine, choline and purine nucleotide and deoxythymidine monophosphate. Now, absorption, transport and storage. Most of the dietary folic acid are found as polyglutamate. It contains 3 to 7 glutamate residue. Duodenum and jejunum, which splits the glutamate residue and form monoglutamate. Only monoglutamate is absorbed. In the intestinal cell, it is again forms polyglutamate and passes into the blood. Folic acid reduced within the cell, principally in the liver, to tetrahydrofolate through the reaction of dihydrofolate reductase. The body can store 10 to 20 milligram of folic acid that will usually last for 2 to 3 months. Dietary sources. Folic acid is widely distributed in nature. The rich sources are green leafy vegetables, whole grains and cereals. The animal sources are kidney, liver and eggs. RDA is around 200 microgram and during pregnancy it is 400 microgram. Function. Tetrahydrofolate is actively involved in the 1-carbon metabolism. Tetrahydrofolate derivatives is to carry and transfer the various forms of 1-carbon units during the biosynthetic process. 1-carbon units are either methyl group, methylene group or formyl group or formamino groups. The 1-carbon unit binds to the tetrahydrofolate at the position 5 and N10 or both N5 and N10 of the pyral structure. Tetrahydrofolate is mostly trapped as N5 methyl tetrahydrofolate, which form is the present in the circulation. Vitamin B12 is needed for the conversion of the N5 methyl tetrahydrofolate to active tetrahydrofolate, where the homocysteine is converted into methionine. This step is essential for liberation of the free tetrahydrofolate and for it repeatedly used in the 1-carbon metabolism. In vitamin B12 deficiency, the conversion of methyl tetrahydrofolate to tetrahydrofolate is blocked. Folic acid deficiency. It is most common vitamin deficiency observed in the pregnant woman. The deficiency may due to inadequate intake, defective absorption and increased demand. Deficiency causes macrocytic anemia. The most pronounced effect of the folic acid deficiency on cellular process is upon DNA synthesis. This is due to an impairment of the purine and deoxynucleotide monophosphate synthesis, which leads to cell cycle arrest in the S phase is rapidly proliferating cells, in particularly hematopoietic cells. The inability to synthesize DNA during the erythrocyte maturation leads to abnormally large erythrocyte termed macrocytic anemia associated with megaloblastic changes of the bone marrow. Deficiency of the folate in pregnant woman has been implicated in neural tube defect in fetus. It is the common practice to prescribe folic acid during the pregnancy and also before pregnancy who wants child. Now vitamin B12 cobalamin. Cobalamin is the only vitamin that is synthesized by neither plants or animals, but only a few species of bacteria. It is therefore absent from all plants, but it is concentrated in liver of animals bound in protein as methylcobalamin or 5'-deoxyadenosylcobalamin. Absorption, transport and storage. Vitamin B12 is present in the diet in a bound form to protein. Gastric hydrochloric acid and pepsin releases this vitamin from protein. Vitamin B12 then binds with a protein which known as cobalamin secreted from the salivary gland. Cobalamin have higher affinity than intrinsic factor of Castle, a glycoprotein secreted from the gastric pyridoxal as acidic pH. In duodenum, cobalamin is hydrolyzed, releasing the vitamin from binding to the intrinsic factor of Castle. From duodenum, cobalamin intrinsic factor complex travel through the gut. This is the flowchart or picture of the absorption of the vitamin B12. The complex bind to the specific receptor on the surface of the mucosal cell of the distal third of the ileum. The binding of the complex and entry of the vitamin B12 is mediated by calcium ion. The mucosal cell vitamin B12 is converted into methylcobalamin. It is then transported to the circulation in a bound form of protein, namely transcobalamin. Methylcobalamin is mostly found in the transcobalamin 1 90% and less degree of transcobalamin 2. The transcobalamin 2 is degraded within the lysozyme and free vitamin B12 is finally released into cytoplasm, where it may be transferred into proper coenzyme by cellular enzymes. Methylcobalamin, which is an excess, is taken by liver and converted into 5'-deoxyadenosylcobalamin and stored in this form. Storage. The total amount of vitamin B12 stored in the body is about 4000 to 5000 or 4 to 5 milligram in adults, an amount sufficient to meet the body needs for 4 to 6 years. Around 80% of these are stored in liver. Approximately 0.1% in this is lost in per day and secreted into gut which are reabsorbed. Dietary sources. Foods of animal origin are the only source of vitamin B12. Liver is the largest source of vitamin B12. Kidney, meat, egg, milk and cheese are also good sources. RDA 3 microgram, in pregnancy and lactation 4.5 microgram. Function. Cobalamin is required in two reactions that leads to formation of the methionine and succinyl CoA. Number 1, homocysteine to methionine conversion. This reaction requires methylcobalamin as coenzyme and catalyzed by homocysteine methyltransferase causes conversion of the homocysteine to methionine. Number 2, formation of succinyl CoA. Vitamin B12 as 5-deoxyadenosylcobalamin acts as a coenzyme for this enzyme methylmalonyl CoA mutase. This enzyme catalyzes conversion of the methylmalonyl CoA to succinyl CoA. Methylmalonyl CoA formed from valine, leucine, methionine, bipropionyl CoA. Fatty acid having odd number of carbon atoms, succinyl CoA may be oxidized into TCA cycle. Deficiency manifestations. Major deficiency manifestations are two types: hematological abnormalities, that is megaloblastic anemia, 25 to 50% larger with fragile membrane and tendency to hemolyze. Number 2, neurological deficiency, subacute combined degeneration of the spinal cord and demyelination of the nerve cells. Pernicious anemia. It is a condition in which deficiency of the vitamin B12 occur due to decreased absorption of the vitamin B12 due to absence of the intrinsic factor of Castle. It is characterized by decreased RBC count, low hemoglobin level and progressive neurological deterioration. It is an autoimmune disease with a strong familial background affecting people over 40 years of age. Antibodies against intrinsic factor of Castle are generated leading to deficiency of the intrinsic factor of Castle and decreased absorption of the cobalamin. Now, folate trap hypothesis. The effect of the cobalamin deficiency are most pronounced in erythropoietic tissue of the bone marrow and the mucosal cells of the intestine. These tissues needed by both N5 and N10 methylene and N10 formyl form of tetrahydrofolate for synthesis of the nucleotide required for DNA replication. The vitamin B12 deficiency, the utilization of the N5 methyl form of tetrahydrofolate is in vitamin B12 dependent. This is the flow chart of the folate trap. Cobalamin deficiency leads to a deficiency of the tetrahydrofolate from needed in the purine and deoxythymidine monophosphate synthesis result in symptoms of the megaloblastic anemia. Thank you all for listening the lecture. Assalamu alaikum.